Hello, I've got a quick one here. I want to talk to you about how we're going to dissect these clams and what structures you're going to be looking for, and kind of give you an idea what they look like so you know what you're looking for. All right. Of course, remember that we're in the phylum Mollusca, and there are several classes, but there are three classes that we're concentrating on because they're so common and so commonly talked about. The gastropoda, the gastropods, those are snails and slugs. The bivalvia, which is what we're going to be dissecting today, those are clams, mussels, oysters, anything with a two-part shell. And the cephalopods, the cephalopoda. And cephalopoda are, the active predators, and the squids, octopuses, cuttlefish, nautilus. Those are also in the phylum Mollusca. So in this lecture, we're going to talk about clam dissection because it has very typical bivalve anatomy, and they're very easy to see some of the structures. And so this is the first time that we've done any hardcore dissection in lab. And so just some general things to go over. I've given you the instructions in homework number 6. And so you can just follow those along step by step. I've also given you some links to some websites that show you step by step, that also show you good pictures. So there's lots of references to help you figure out what you're doing. You want to wear gloves and goggles. You don't want to splash stuff into your eyes. These things are preserved in a very benign preservative, but you still don't want to get anything in your eyes. And there could still be bacteria and things like that. The gloves, same deal, and the gloves will help you with your grip too, help you clean up. You're going to be using sharp instruments, all right? And these things are going to be stored in wet preservative, so they have the possibility of being a little slippery in that. And you're also going to have people all leaning in close to one another. So you want to be very careful, all right? You're adults here, you know what you're doing, but just be mindful that you got slippery surfaces and slippery hands and sharp instruments, and so just pay attention. Now, one thing that students, when they first dissect, a lot of times they tend to overuse their scalpel. They want to scalpel everything. And actually the tissue scissors, the little scissors, those are kind of your go-to instrument. Those are the best thing for being able to dissect. And you won't use the scalpel as much, but in this dissection, you actually do need that scalpel. You need a thin, sharp blade to open up the clam. When you're done, cleanup is pretty simple. This stuff is all benign. It could just go into the trash. But of course, clean off your instruments, clean out the pans, wipe down the table. Please don't leave a mess. All right? So in all the dissection instructions for today and for future dissections, I always try to give you proper direction, like this is anterior to that, or look at the ventral surface, look at the dorsal surface. So you got to really know your way around the organism. You got to know up from down and left from right and front from back, right? You need dorsal, ventral, anterior, posterior. Well, that's pretty easily on things that have cephalization, right? That have a head, but bivalves don't have a head. So how do you tell which end is anterior and which end is posterior? In the clam and in other animals, when you're trying to figure this out, think about which way they move. They tend to move forward toward the end, and that should tell you what the anterior part of the animal is, right? And so when you think about the clam, think about how the clam moves, and that will tell you what is anterior. So we'll talk about this in more detail in a second. Okay, we always start by checking out the external anatomy, the stuff you can see without cutting. Not a lot on the clam. The shell, the hinge, and the umbo is what you need to know. But I also want you to, if I show you a picture of a clam, you got to tell dorsal from ventral, anterior from posterior. And so the shell, of course, is just the hard outer shell. The hinge is what holds the two pieces of the shell together, and it's kind of a ligament. It's got some elasticity in it. And the umbo is just sort of that beak, that sort of horn at the top. You can also see growth rings on here, and so you could count those rings, and you could actually age the muscle by counting those rings. And so Dorsal and ventral are pretty easy in the clam, right? The umbo, the hinge, that's going to be dorsal. The clam is going to be stick itself down into the substrate, and so that would be the ventral surface down in the substrate. The dorsal is what sticks out of the substrate. Anterior and posterior are not super easy to tell when you're looking at the shell. What you need to look at the foot. Again, think about how the clam moves. It sticks out that muscular foot, it kind of fills it with blood, or it sticks it out, then it fills it with blood to make it swell, so it kind of wedges in the substrate, and then it uses muscles to pull its body up to the foot. And so stick out the foot, wedge it, pull the body up. So the foot is going to indicate the anterior portion, the front of the clam. And so, again, from the outside, it's not always easy. Usually you have to see the inside. And in this clam, this is a commonly used specimen, so you sort of know, shaped on the umbo, kind of where the foot's going to be. But really, you got to see that foot. Okay. Stuff you need to know for the internal anatomy. Once you cut it open, these are things that we want to identify, we want to get pictures of. If I show you a picture of it, I want you to be able to identify it. The adductor muscles and their attachment points, the mantle, the incurrent and the excurrent siphon, the foot, the gills, the labial palps, the mouth, stomach, intestine, digestive gland, gonads, heart, and the pericardial cavity. And we're going to look at all those here in a second. Also, know the function of those, and you know the function of most of these things, but some of the structures like, the mantle, the labial palps, we haven't seen those before. Know what they're used for. Now, some of these structures in the clam are not obvious, okay? And so it's not really always easy to see them, but you at least need to know where to look for them. And so for those kind of structures, I may not ask you to identify the structure, but I may ask you, hey, where would you look for this structure? And again, if you know the function, you should be able to figure out where to look for it. Okay, and so this is just a general diagram of the clam internal anatomy. Now, my suggestion is when you dissect your clam is to make it mimic this picture, and that way it's much easier to find everything. And so what you want to do is leave all the viscera in the right half of the clam. So what we're looking at here, if you look at anterior and posterior, then you realize we're looking at the right half of the clam, okay? And so if you cut your clam and leave it and just look at the right half, then it'll match up to this diagram and it'll be a lot easier to find things, right? If you had the left, everything would just be reversed. Now, what's even easier is start like this, but eventually you want to, just like you're shucking an oyster, you want to just scoop all of that, the viscera out of the right hand and then have just the viscera, and then you can pick them up and move them around. You can find stuff a lot easier, but keep yourself oriented to this picture. Okay, so first thing we talked about were the adductor muscles. These are muscles that pull the shell closed. And you've got an anterior and a posterior one. And to get your clam open, you're going to have to cut these. So one thing you'll notice once you start to study these is they are very tough, and they're quite big, quite large muscles, and they're very short, so they can develop quite a bit of force here, and that's why it's real easy. I mean, the clam can close up, and it's very hard to open them up because these muscles are so strong. Well, the clams we're dissecting, it's the same thing. They're preserved with the shell shut, and these muscles are so strong you just can't force that shell open. It's just not going to happen. You'll break the shell long before you open it. So you've got to cut these muscles. So look at this structure and picture where these muscles are. They're a little bit dorsal. They're closer to the hinge. And there's one on both the anterior and posterior. And so what you're going to do, this is why you need your thin scalpel, because that's the only thing that'll fit between the shells of the clam. And you want to slide it along until you feel something very hard and kind of rubbery. That's going to be it. So then use that, and you need that sharp scalpel, and you just cut right through that. And then that'll loosen it a little bit, then go to the other one and do the same thing. And then you kind of go from one to the other, back and forth, and keep cutting each time, and it'll get looser and looser, and then you'll be able to pop it open. And so this is kind of what I'm doing here showing how you see this is now with this clam is upside down, the ventral's pointing up, and I'm cutting down. And again, when you're cutting with things, especially when your hands are wet and that, cut away from you, cut down so you don't cut and stick, your knife doesn't slip and you stick it in your gut. So you see how I'm cutting down toward the dissecting pan. But right about there is where I'm hitting that. And again, a new scalpel blade that's very sharp makes a huge difference here. Once you get it popped open, then you should be able to see the muscles and where they attach to the shell. And so what I'm pointing at here is one of the adductor muscle attachment points, and you can see the other one sort of opposite from it. And so poke around on that. See how tough that muscle is. See how solidly it is connected to that shell, and you can see why they can pull that shell tight and keep it tight. Okay, so then on the inside of the shell, this kind of skin that runs all around the inside of the shell and sort of surrounds the viscera, that's the mantle. And that's a structure unique to the mollusks. And the mantle kind of acts like a pouch to hold in the viscera, but its most important function is it secretes that shell. And so the cells of the mantle secrete the hard shell and build that hard shell. Of course, this is also where like pearls form. So if an irritant gets in there, the mantle can grow around the irritant to sort of trap it, to keep it from getting into the clam. But as it does that, it covers it with layers, and that's how you make a pearl. And so this, thin skin-like stuff that should be easy to remove. And again, if you've ever shucked an oyster, you kind of stick a tool in there and just kind of go and scoop all the stuff out. What you're doing is you're loosening the mantle from the shell. Okay, next thing we want to look at are the in-current and the ex-current siphons. And so these are just tubes that, in-current sucks water in and ex-current blows water out, right? So this is a sedentary organism that filter feeds, so it needs a way to suck in water and get rid of water. Now in this diagram and in lots of diagrams, they'll look like tubes. And in a lot of species, it's like a tube, you can see it. But in the species that we dissect in lab, they're not really tubes. They're more just sort of openings. They're not as obvious. But again, this is one of those where you know where to look for it, and then you can figure out what you're looking at. All right? And so they're just kind of an opening. Now, no matter what the species, the in-current is always ventral, and the ex-current is always dorsal. And so here we're looking at the posterior end of the clam, and this is the ex-current siphon. So you see it's not like an obvious tube like was on the diagram, it's just an opening. But this ex-current is always dorsal to the in-current. Again, think about the in-current is bringing in fresh water that's got oxygen and is also bringing in food. So that wants to hit the gills first. The ex-current is getting rid, that water is, you've used up the oxygen, you've got waste. And so the ex-current siphon, you want to blow out and get rid of that stuff. And so what I'm pointing at here are one set of gills. And again, where the gills are, that's going to be the in-current siphon, because you want that fresh water to hit the gills first. Okay, now the real obvious structure is the muscular foot. And again, the foot defines the anterior end of the clam. And once you have the anterior and posterior, then you can figure out left, right. So you realize that in this figure, anterior is to the left of the diagram. And so if that's the front of the clam, you stood the clam up, then you can figure out, oh yes, I'm looking at the right half of the clam in this picture. And so the muscular foot's pretty easy to identify. It's very tough. It's it looks like this. It looks like just like a very solid foot. Another way to describe it. But the texture of this is very obvious. Very obviously a muscle. And that defines the anterior end. Now, opposite the foot, at the posterior end, are the gills. And again, they're going to kind of define where the in-current siphon is. And sometimes when you're pulling your clam open and you're moving the viscera around, the gills, they're so delicate and light that they kind of get folded over, they might get ripped, but you can kind of figure out where they should be relative to the foot, and again, they'll help you find the siphons. And so, you know, everything here kind of, because in these preserved specimens, you know, has the same color. Now in a live specimen, they would be probably more red, right? There'd be yeah, they would have a more reddish tint anyway. I don't know if that's true. I should back up. Now here's a picture showing the gills. Again, everything's sort of the same color. You kind of go by texture. The gills are very delicate, and they're on toward the posterior end, sort of. Now there's a pair on either side. So if you carefully tease these apart, you can see that there's two gills here, but then underneath the viscera on the other side, there's also another pair of gills. Again, where the water gets sucked in. Now these gills are held in a special cavity called the mantle cavity. And so this is not a typical cavity. I think, again, this is unique to mollusks, but it's an opening just for the gills that's formed by the mantle. Most organisms, most animals don't have this. So the water then runs in over the gills. Of course, there's gas exchange takes place, but also the gills produce a mucus and act as a filter. And so they filter feed. They trap things, little things in the water in that mucus. And then that mucus just gets kind of worked toward the front, toward the anterior end of the clam. As water moves in, as the gills make that mucus, it just gets kind of worked. Again, probably the muscle of the foot kind of helps a little bit. That mucus gets worked to the front, and then it needs to get into the stomach. And so, you don't really have it's hard to see a mouth. The stomach and mouth are very hard to see in this organism, but you can see the labial palps. The labial palps are just two big flaps on the anterior end. Again, usually where's the mouth most times? It's at the anterior end. And those labial palps just kind of help gather that mucus and push it into the mouth. And from there it goes to the stomach, and from the stomach through the intestine, and then the intestine dumps back into the excurrent siphon so you can get rid of the waste. Now that makes a complete gut. And so this is the first phylum we've seen with a complete gut. In our other phyla, we've had just an open gut. Everything, there's only one opening, right? Stuff comes in and stuff goes out in the same opening. Now we're seeing more modern organisms, and you've got this complete gut, which allows the food to enter one end and the waste to leave in another end, which can be more efficient, potentially. And so if you cut off the gills, get them out of your way, a lot of times that will expose the labial palp. Again, there's two, one on either side. So once you find them, you can kind of see where the mouth should be. And again, the mouth is one of those things, it's not easy to see, but you know where to look for it. But the labial palps you can see. And here you go, they're big flappy things, and in between them is the mouth. So if you poke around with your probe, you ought to be able to find an opening, and that's going to be the mouth where the food goes in. Now, what's interesting, in this clam, in these mollusks, this is where we're first starting to see a coelomic cavity, right? Our earlier organisms, either didn't form a mesoderm, so there's no coelomic cavity, or they formed a mesoderm like the flatworms, but they're acoelomic, they don't form a cavity. Now we're starting to see organisms that form a cavity, a coelomic cavity where the organs can get tucked into. But this is an early version of that, so it's not well developed. So in the clam, the coelomic cavity just has the heart. But the stomach, the intestines, all that stuff, the digestive gland, the gonads, they're still not in a cavity. Like you're used to having all your guts in a cavity, right? And so the stomach and the digestive gland and the gonads, they're all sort of tucked in to the middle of the foot. So you got this big muscular foot, and there's not really a cavity, but just at the top of this foot is a solid mass where all these things are tucked into. So the digestive gland is akin to the liver. It produces digestive enzymes, and it's got a different texture, and it usually has a greenish color. The gonads are not well defined, like you know, in a lot of organisms you can identify the gonads. In this, they're just kind of, again, a different texture. But to find it, you know, there's no cavity to cut open. So what you got to do is cut off part of the foot. So if you cut off the ventral part of the foot, the muscular part that would stick in the substrate, cut that part off, and then kind of turn it up, and then make a sagittal cut. So the sagittal plane, you know, that creates bilateral symmetry. If you cut along that and you sort of butterfly it. So if you've ever butterflied a pork chop or something, where you cut down, you know, you've got something and you cut along the thin edge and you peel it back, and it opens it up like a butterfly. That's sometimes a good way to be able to see these structures, but this is difficult. These structures are difficult to see when you dissect, and so the better you are at this, the better you'll be at seeing these things. And so here's an example of what I call butterflying. So this is a ventral view, where you've kind of gone up the sagittal plane and then peeled that foot open like a butterfly. And if you do it right, you can see the digestive gland, which is up near the mouth, that's got a different color. You've got this funny texture stuff, that's gonads. The stomach is sort of just not really well defined. And you can often see that intestine. You'll see that tube running through there, and that's the intestine. Like I said, they're not inside a cavity per se. So again, this is the first coelomic cavity. But since it only contains the heart, we call it the pericardial cavity. And you've got a heart, but still an open circulatory system. So the heart has got, you know, kind of a valve, a vessel that runs into it and a vessel that leaves, but then that's it. When you follow them, they just sort of dead end, and then the blood just kind of swishes around. Okay? And so that's going to be the dorsal portion of the clam. And an easy way to get at this is, again, to pull all the viscera from the shell. So you got rid of both shells, and you're looking at the viscera, and then set it upright like the clam would sit, you know, so the dorsal surface is pointing up, so you're looking down on it, and the heart's going to be right there, but there's a thin membrane that sort of covers it. And so if you can peel that membrane away, then you can see that cavity, that pericardial cavity. The heart will be right there. That's the easiest way to see the heart. And then once you've observed the heart, got pictures of it, you can pull the heart out, and then you can see the bottom of that coelomic cavity. Often you can see part of the intestine. And so here's a dorsal view. So if you can imagine, I took the viscera, shucked it out of the shell, and then stood it up. And now I'm looking, this is like if I'm standing over the clam and looking straight down. And you can see that thin membrane I've pulled away, and then you'll see this muscular heart, and you've got kind of a blood vessel coming in and one leaving, and there you go. Now you pull that heart out, and at the bottom, I think that dark stuff is kind of like, sort of acts like kidney tissue a little bit, early kidney tissue, and you can often see that intestine running. And again, think of where that intestine's going. It's heading back to the excurrent siphon. And so here then is a lateral view. That was a dorsal view. So now again we're back to looking at the side, but this has been perfectly bisected, and you can see that cavity. You can see number 7 is that coelomic cavity, or the pericardial cavity, and then the heart hanging out in that cavity. And then you could imagine that intestine running along the bottom of that coelomic cavity, and you can kind of see it here where it dumps into that excurrent siphon. And so you can see other structures here that you need to know: the mantle, the gills, the anterior and posterior adductor muscles, the hinge, the foot, the digestive gland, and the gonads. So this is, if you could make your clam look like this one, this is a very excellent job here. But you can see how this time, to get at the digestive gland and the gonads, they didn't butterfly it. They just sort of took their scalpel and just ran along the sagittal plane and just completely bisected it. And that seems to work nicely too, if you could pull that off. Okay, so that's all the parts to the clam. So you've got the instructions. Let me know if you got any questions. See ya.